COVID-19. COVID-19, MERS, and SARS. In January 2020, a novel coronavirus, SARS-CoV-2, was identified as the cause of an outbreak of viral pneumonia in Wuhan, China. The disease, later named coronavirus disease 2019 or COVID-19, subsequently spread globally. In the first 3 months after COVID-19 emerged, nearly 1 million people were infected and 50,000 died. By 6 months, the number of cases exceeded 10 million, and there were more than 500,000 deaths. One of the troubling observations about COVID-19 is that people who are infected with SARS-CoV-2, some scientists estimate up to 40%, can transmit the virus to others before they have symptoms or without ever having symptoms of disease. The Centers for Disease Control and Prevention, or CDC, developed a test to diagnose COVID-19 in respiratory and serum samples. The NIAID also is accelerating efforts to develop additional diagnostic tests for COVID-19. These tests are helping facilitate preclinical studies and aid in the development of medical countermeasures. NIAID COVID-19 research efforts build on earlier research on severe acute respiratory syndrome, or SARS, and Middle East Respiratory Syndrome, or MERS, which also are caused by coronaviruses. MERS is a viral respiratory disease that was first reported in Saudi Arabia in September 2012 and has since spread to 27 countries, according to the World Health Organization. Some people infected with MERS coronavirus, or MERS-CoV, develop severe acute respiratory illness, including fever, cough, and shortness of breath. From its emergence through January 2020, WHO confirmed 2,519 MERS cases and 866 deaths, about 1 in 3. Among all reported cases in people, about 80% have occurred in Saudi Arabia. Only 2 people in the United States have tested positive for MERS-CoV, both of whom recovered. They were healthcare providers who lived in Saudi Arabia, where they likely were infected before traveling to the U.S., according to the CDC. Infection with SARS coronavirus, or SARS-CoV, can cause a severe viral respiratory illness. SARS was first reported in Asia in February 2003, although cases subsequently were tracked to November 2002. SARS quickly spread to 26 countries before being contained after about 4 months. More than 8,000 people fell ill from SARS and 774 died. Since 2004, there have been no reported SARS cases. Research evidence suggests that SARS-CoV and MERS-CoV originated in bats, and it is likely that SARS-CoV-2 did as well. SARS-CoV then spread from infected civets to people, while MERS-CoV spread from infected dromedary camels to people. Scientists are trying to determine how SARS-CoV-2 spread from an animal reservoir to people. Characterizing MERS, or MERS, and COVID-19 disease. Since the emergence of MERS coronavirus, or MERS-CoV, in 2012, and SARS-CoV-2 in 2019, NIAID scientists have advanced their understanding of how the viruses can cause disease, focusing on developing animal models of disease and on countermeasures such as diagnostic tests and vaccine candidates. Research by NIAID scientists and others demonstrated that MERS-CoV and SARS-CoV-2 use their spike or S protein to enter cells and initiate infection. After entering the cell, the viruses delay unusual immune system responses, allowing the infection to gain a foothold. By the time the immune system responds, the infection has progressed and becomes much harder to fight. Scientists also have characterized different strains of MERS-CoV and can determine through tests how those strains affect different animals. They also are studying how the stability of the virus changes in nature depending on the temperature and humidity, where it exists, and whether its form is liquid, droplet, or aerosol. Using a monkey model of MERS, researchers have learned how the virus causes disease in people. For example, scientists at NIAID's Rocky Mountain Laboratories, or RML, demonstrated in a monkey model that clinical signs of MERS appear within 24 hours of infection. They also found that the virus causes disease deep within the lungs, leading to pneumonia. NIAID-funded researchers also have established several mouse models of infection that have been critical in developing MERS-CoV medical countermeasures. A research group at RML also has developed a rhesus macaque model of SARS-CoV-2 to study COVID-19. The model mimics mild to moderate respiratory disease in people, including signs of pneumonia seen in an X-ray. This also is an important diagnostic feature in human patients. They also learned that the virus causes disease in the lungs and that virus is shed from the nose, throat, and rectum in a pattern similar to virus shedding in COVID-19 patients. The scientists are using this model to evaluate treatment and preventative vaccines. Characterizing MERS and COVID-19 disease. Since the emergence of MERS coronavirus, MERS-CoV in 2012 and SARS-CoV in 2019, NIAID scientists have advanced their understanding of how the virus can cause disease, focusing on developing animal models of disease and on countermeasures such as diagnostic tests and vaccine candidates. Research by NIAID scientists and others demonstrated that MERS-CoV and SARS-CoV-2 use their spike S protein to enter cells and initiate infection. After entering the cell, the viruses delay usual immune system responses, allowing the infection to gain a foothold. By the time the immune system responds, the infection has progressed and becomes much harder to fight. Scientists also have characterized different strains of MERS-CoV and can determine through tests how these strains affect different animals. They also are studying how the stability of the virus changes in nature depending on the temperature and humidity, where it exists, and whether its form is liquid, droplet, or aerosol. Using a monkey model of MERS, researchers have learned how the virus causes disease in people. For example, scientists at NIAID's Rocky Mountain Laboratories demonstrated in a monkey model that clinical signs of MERS appear within 24 hours of infection. They also found that the virus causes diseases deep within the lungs, leading to pneumonia. NIAID-funded researchers also have established several mouse models of infection that have been critical in developing MERS-CoV medical countermeasures. A research group at RML also has developed a rhesus macaque model of SARS-CoV-2 to study COVID-19. The model mimics mild to moderate respiratory disease in people, including signs of pneumonia seen in an X-ray. This also is an important diagnostic feature in human patients. They also learned that the virus causes diseases in the lungs and that virus is shed from the nose, throat, and rectum in a pattern similar to virus shedding in COVID-19 patients. The scientists are using this model to evaluate treatments and preventative vaccines.